series brought to you by the Dauphin Island Sea Lab, the Alabama Aquarium at the Dauphin Island Sea Lab. Today we are streaming live from the RV research vessel Pelican. We are going to tell you a little bit about this morning. This is with the Hellenic Center for Marine Research in Greece. He is our international collaborator on this project, and he's going to tell you very broadly what kind of about what we are examining during this project.
Great. Greetings from the Gulf of Mexico. We are offshore from the Louisiana, and we are cruising in this area in the last 4 days, and we are here in order to investigate one of the most important cycles in the Earth's surface. Basically, we are looking at the cycle of silicon, which is the second most abundant element on the Earth after oxygen, and still in 2024 there's a lot we don't know about it. So the main objective of this cruise, this project, is to try and identify any controls that microbes have on silica, especially after the sediment is deposited on the bottom of the ocean. We know for many years that microbes have a lot to do with what's happening in the water column with diatoms, and they accelerate the dissolution of diatoms, which are phytoplankton, comprised of silica. But we don't know anything about if there's any involvement of microbes after the material is deposited in the bottom of the ocean. After some initial experiments that we did a few years ago, we realized they have a major influence, and the main purpose of this cruise is to try and quantify the influence, and also identify the role of microbes, exactly what they're doing, how they're doing it.
Could you explain some of the ways that silica and silicon are very important? You mentioned that they are important. Can you describe some of the reasons they're important?
There are different parts of the silica cycling in the Earth. One of the main ones is what's happened on silica on land. And basically what's happening is that silica is a major part of many minerals, that with the influence of CO2 in the atmosphere, minerals get weathered, and they release silica into the streams, into the rivers, which eventually comes into the ocean. During this process on land, a lot of CO2 is consumed from weathering these minerals, and this is one of the main controls of the CO2 cycle in the atmosphere. If that CO2 will be so high that will be the planet will be uninhabitable. Now, once the material enters the sea, there's another set of processes that are happening, which is basically the reverse of what has happened on land. So basically, silicon is incorporated into new minerals in the bottom of the sea, and that, as a result, releases CO2. So it offsets a bit of the consumption of land. So we have a cycle that basically consumes CO2 on land and returns it into the sea. And so that's why we have a relatively stable CO2 level in the atmosphere.
And it used to be thought that the storing of the silica in minerals, at the bottom of the ocean, that's a long time, and recent evidence suggests that is happening faster than previously thought, right?
Yes. First, it was thought that it didn't happen. So that was 30, 40, 50 years ago. Then gradually we did some experiments of the Amazon River and then in the Mississippi that show that this uptake and the formation of the new minerals is pretty quick, which means that it happened in the order of months to a few years, which is for geological timescales is extremely fast. Then recently, with the use of radioisotopes like silicon-32 that we are using in this cruise, we found out that this is even more rapid. That can happen in a few hours after we sample the sediments. The whole process starts, and it starts having significant influence on the levels of silica, both in the water, decrease in the water, and we see a corresponding increase in the particles, which means that within a few hours, silica starts being incorporated into minerals.
Role of the silica cycle with the CO2 in the atmosphere. You also mentioned the phytoplankton diatoms, and you mentioned that the diatom, so the diatom structure, they have kind of an external test. It's like a shell that's made of silica, and the diatoms they produce oxygen, and they produce a significant portion of the oxygen on the planet. And so this silica cycling also affects the and through the diatoms it has the potential to impact more of our atmospheric.
Yes, diatoms are among the most important primary producers, which means that they are photosynthesizing at the surface of the ocean. So they suck up CO2 and they release oxygen. The influence, and this is basically on a steady state, which means as much oxygen as they produce, they also consume during the decomposition. There's a tiny fraction of that organic matter that they produce during photosynthesis that escapes decomposition. And once this material in the form of diatoms or any kind of organic matter ends up being buried in the sediments and escapes decomposition, that tiny fraction of organic matter is has an equivalent in oxygen that accumulates in the atmosphere. So basically, whatever we find in the sediments in the form of carbon that has formed during the photosynthesis that escapes further decomposition, it translates into the amount of oxygen we find in the atmosphere. So it's both important for the carbon cycle, for the silica cycle, and for the oxygen cycle.
So deploying mainly two different types of instruments. So we're going to show you the instruments that we're using to collect these samples. This one is called a multi-core, and we can kind of describe it to you set up that way, and then we'll show you one of the cores over here, or the core tubes. We don't have any mud in our tube currently, but this is a multi-core. It's set up to take 8 different core samples of the bottom, and it is a mechanically triggered device. This is dropped on a cable, and you can see the weights there, and when it hits the bottom, the cores, the clear tubes, or the tubes are driven down into the sediment surface, unless it's too hard, as it was this morning when we at our first station for the morning. And then it's spring loaded, so this will drop, releasing this cap that puts a suction on the tube, hopefully with some sediment in it. This arm right here is released, dropping this foot that holds the mud, the sediment, inside this clear tube. And then we can take these tubes, we've got 8 different cores, and so those are being sampled by different labs, taking lots of different samples, and then they're processing those samples to get different kinds of data. So Claire, are you available to kind of show how this would be moved, the tube would be moved over to the extruder?
Yes, I can. I might need help opening it.
It is strong spring, so it can be hard to release that. And you might have to hold the tube, and then it goes to an extruding device where we can, so if you imagine that this clear tube had mud in it, and then sometimes it also has like water that would have gone into the tube from the from the bottom next to the sediment layer on the bottom, then we would push the mud up by pushing this core down, and so then we can collect layered samples of that mud. So Katie, you want to kind of tell folks about, this is Katie Howe. Katie is a postdoc at the Dauphin Island Sea Lab in Dr. Kielreese and Dr. Krause's labs. They're co-PIs on this project. Do you want to kind of explain to people how you would take your samples and what you're doing with them?
Yeah, sure. So we would go ahead and take this part off, so it was just the tube, and as you push down, you get a layer here, and then we just use a flat piece of metal to cut it so it looks like a hockey puck. And then we take those pucks and we put them in plastic bags, and we take some samples from them.
Can you grab that little tub? Because let's kind of give folks an idea of the kind of sediment we're getting. It's not sandy like you'd expect to see at the beach. And if you could like maybe smear it, it's like a fine clay.
And so Penny, if you want to come over. Since we work with biology, we use a lot of protective equipment, including masks and gloves.
Because it would affect your sample results?
Yes, we don't want any of the bacteria from our hands to get onto into our samples. And so here we have a tube that's pre-filled with formaldehyde, so that when we put the sample in, it gets preserved right away.
So just to clarify, they are not, they're just doing a simulation of collecting a sample, because we did not have a, when we're back here actually doing the sampling, it's pretty hectic, so it would be hard to do a live stream while we're all back here working, but we're kind of simulating some of the sampling techniques to demonstrate.
And so we have our little piece of sediment in the formaldehyde, and then we just shake it so that the formaldehyde preserves all of the cells that are in there. And then we would freeze the rest of the core to do DNA and RNA extractions later on in the lab.
This is Did I say that right?
Yes.
Penny is a PhD student in Dr. Kielreese's lab. And that is from the University of Colorado at Boulder. And she is here with Dr. Shoily Rahman, one of the collaborators on this project. Thank you, Claire, for helping us demonstrate how a core works, how extruding a core works. Dr. Rahman? Would you like to collect a sample? Dr. Rahman takes several different kinds of samples from these cores.
Sure. So if you imagine the core back here on this extruder, each of these holes right here, I'm not sure if someone demonstrated this already, they are marked at 1 centimeter depth intervals so that I can measure precisely what section of the core or what layer I'm sampling into. So then if you can imagine that I've extruded some of the sediment, it's exposed, so then I'm going to take some of this and put it into, very carefully, and I should have gloves on, a centrifuge tube. So it's this conical vial, and then I'm going to spin it at around 3,000 to 4,000 RPMs, that's rotations per minute, so that I can, the fluid that's in here will rise to the top, and the sediment will be compacted because what I'm sampling is that fluid that's in this layer of sediment. For trace metals, for heavy metals, I also look for arsenic and cadmium, so things that are toxic to humans and also maybe magnified in your food chain, like our food chain, if you like.
So some of the other, this is not the mud. There's like the whole 14-person science party is usually back here collecting samples, and so we're also measuring oxygen through the core, the length of the core from the surface sediment down. We're also collecting samples to look for lead and a number of other from these sediment cores. One of the processes that is being examined here is the role of oxidizing metals, so metals in the sediment that interact with the falling organic matter from plankton and other things, and through that release carbon dioxide in this process that's known as reverse weathering. So we are going to go around the, do you want to show your filtration there, or do you want to show the CTD? Where it comes from.
We can do the filtration. So this is, and I have several cubitainers of this. This is a 20-liter cubitainer.
I'm going to have about a show around the corner.
Sure.
That instrument over there. Lacey, could you point to the rosette? So the water by that instrument.
Yep. And we are filtering it through this is coated with manganese oxides, and we are looking for a radioactive element in here naturally occurring, which will give us some measure of how much, when we're collecting these cores, how much of this fluid actually gets released into the water column. So it gives us an estimate of how much water in just the sediment and the water column. So if I measure arsenic in this Poral water and I have an estimate of how much is exchanging, then I can tell you how much is being released into the water column and may be available for fish or for humans. Yeah.
So it kind of gives you an idea of whether some of these compounds are kind of staying in the sediment or are moving through the water column to different depths.
That's correct. Okay. You want to show them?
So we will go over to the other instrument that we've been using to sample. This is do you want to describe the CTD rosette?
These are, Jeff, do you want to describe them?
Say hi, Jeff.
Hi there.
Here. You want to? Yeah.
All right.
The sensors, pistons.
Yeah, so this is kind of an Achilles heel of blue water coastal oceanography, but it's a way for us to collect samples at a very specific depth and also tell a lot of information about those depths. The instrumentation is down here. CTD stands for conductivity, temperature, and depth. C in conductivity is how we get salinity, because the more salt in the water, the more conductive it is. Temperature is just a thermistor, and depth is a pressure sensor. And then you make little corrections as you go really deep for pressure. There's also a suite of other sensors on here, oxygen meter, there's some fluorometers which measure basically light that's given off by organic compounds. Some of those lights, some of those compounds could be things that are dissolved in the water. Think like the color of organics in your tea. Others can be phytoplankton, so the primary producers of the ocean. Their pigments fluoresce specific wavelengths of light, so we can use that to see. So we get all this information as everything's going down. These bubbles are open, lets the water pressure.
And then as we're going back up, we can look at those specific depths of interest, and we can close the bottle. So there's a spring in here. It's coated with, like it can be like a bungee cord, and then there's a mechanism up here that's a magnetic trigger. It's pretty cool. So you shoot a pulse of a pulse of electricity down here, and it triggers this thing to flip up. These are engaged right now, so they're not. But when it flips up, the bottle, and it closes the top and the bottom. So we don't want to disturb it. But this also is a sensor for light that's hitting the light that's coming in from the surface, and then we can measure how the water attenuates that light, or the water kind of the light diminishes in the water.
Collected by the sensors, it goes directly to a computer that will output it in basically real time, and we sit around and watch the profiles appear on the computer screen, and then you can also use that information to decide where you trigger the bottles to collect water samples, right?
Yeah, a lot of the times we don't know where we're going to grab water with this system, and this is a kind of a mid-sized system. Larger systems could be 24 bottles, and the whole setup could be on the order of 3,000 pounds, which is pretty impressive to get this thing in the water. And then larger systems like this and larger vessels can go all the way down to the bottom of the ocean. Some of these sensors aren't rated for that, so if you go that deep, you got to take certain things off. Otherwise, you're gonna destroy them. But it's pretty amazing. You can see, maybe there's a layer of phytoplankton that you didn't realize was there because you can't see 40 meters down. So you want to grab a bottle there, and then you can do it at a variety of interest points depending on what your interests are. So we're in the water. I like to look at diatoms, so those are phytoplankton. But if you're a chemist, you might want to look at specific things down in the deep water where you're trying to understand how the sediments influence the deep water processes, things like that.
Can you show? I think we've got some folks who are ready to show us how we would take the water samples out of the bottles, and
Absolutely.
so we might do it in different ways. So we've got some that would be filtered. The Niskin bottles. So Claire is doing a rinse on that before you put water in it, just to make sure that there's no contamination. So she's using a syringe to draw water, and then Claire, are you going to put that directly through a filter?
Yes, I will. I have filters in the caddy, and I'll attach it to the top of the syringe and filter it into a centrifuge bottle tube.
And so with that, when you process that sample, what is it that you'll be looking for?
We'll be looking at nitrogen, but I can't remember off the top of my head why right now. So if you ask my advisor, she'll probably know.
Hi, so we're looking for dissolved nitrate, and nitrate is an essential nutrient for us for phytoplankton, but we're looking at actually the isotope signature of nitrogen in that nitrate, because it'll tell us whether the nitrate here at this location is coming from, say, agricultural sources, or if it's just from biological processes. So it's a way of distinguishing terrestrial fertilizer input into the Gulf versus natural biological activity.
So Claire just started in Dr. Ramon's lab about 2 weeks before we started the cruise. So she is learning new sampling techniques and research protocols.
On-the-job learning.
So what bags?
The giant...
Your big bag.
Oh my... And I am collecting a bunch of sediment, almost 2 kilos of sediment per layer, and I'm looking for this very rare radionuclide, silicon-32, that's tagged onto this group of phytoplankton to see how much of it persists in sediment and how much of it is transformed. So it's just another measure of seeing how much of this essential nutrient comes out of sediment into the water column and is available again for phytoplankton to use. And those phytoplankton are usually at the base of healthy marine food webs. So knowing this ratio of nitrogen to silicon is really useful for predicting, say, harmful algal blooms versus diatom production. Yeah.
So Dr. Krause's lab is also from these Niskin bottles. He mentioned that one of his areas of research is the diatoms, the phytoplankton, and their part in the silica cycle. And so this is Kayla Barnett, one of Dr. Krause's grad students. She has been taking water samples in bottles. So, Kayla, I don't know if you're doing any sampling from this. You've already taken your samples. So, bottle, you rinse it, fill it, and then what do you do with it?
Typically, I have 5 of these bottles. We choose the different depths based off that CTD profile. I specifically look at ecofluorescence. So reading for chlorophyll. It's about a 1 liter bottle. I take them inside and I have a filtering manifold, which is basically just multiple. And I will dump the water. On the depths, typically lower depths, we do take more sample just because there's not as much ecofluorescence down there. We're going to use that for analysis and chlorophyll A analysis, which is the one. And we're going to be using that for nutrient analysis. Super exciting stuff.
Kayla is also a new grad student. She is in Dr. Krause's lab, so this is definitely immersion learning aboard a research vessel. So is there anything else that y'all would like to point out or add about the CTE or deck operations before we kind of move into the lab?
Absolutely. So there's this big, ominous-looking container. This is a mobile laboratory, but this is one that's specially designed for radiation. And the reason you want to separate radiation, well, there's some obvious reasons. The less obvious is the potential for contamination of natural radiation that certain scientists are trying to measure in the field. So in here they have a sign that says for carbon-14 analysis, carbon-14 is a natural carbon isotope in the atmosphere, that the signals that researchers are looking for that are natural are like looking for one brown grain of sand in 10 dump trucks of tan sand. So very small signals, parts per trillion. And what this means is that anybody that's using, let's say, carbon-14 to do tracer work, where we add carbon-14 to understand how fast things are growing, we can find it to these vans. Now I'm doing the same with an isotope of silicon, and Dr. Rahman is looking at natural abundance of that, so we're keeping our operations very separate, and because we don't want to have contamination of her samples with my material. But these other things, these other labs can be, you can have labs. Notice the boat's made of steel, and so anyone trying to measure iron, it's the iron, all the measuring trace metals like iron, like nickel, cadmium, zinc. Lots of these different types of irons are very easy to contaminate, and so there's special systems that people develop, and then they take them into special trace metal clean labs. You might have seen scientists or people in like computer technology get into these kind of very elaborate suits in order to kind of keep contamination to a minimum. You can use containers like that for those purposes as well.
So can we move into the lab, or did you want to add anything while we're still out?
Not really. These are 2 new graduate students in my lab as well. Say hi. Kendall and Priya. They're PhD students, whereas Kayla, who you met, is a master's student.
Okay.
All right. Here, you can go.
Into the lab.
So welcome to the main lab of the RV Pelican. This is actually a pretty small boat for the types of research that we normally do, but it's laid out exceptionally well for the types of things that we want. You notice they have all these drawers and cabinets, and then we use the bench space. And the way we use the bench space, the way we use this space is very different from how other science parties will do it. In fact, after we get back 2 days later, this vessel will go out with a completely different science party, likely doing completely different things. And this boat does a lot of operations for industry as well, so the types of science that's done on the Pelican is pretty broad and expansive.
Can you point out some of the stations on the bench and what sampling is being done?
So, Katie, our postdoc, and Penny, PhD student, are working over here. They have this nice tape barrier. Basically, we all stay away from that because they're looking at biology and molecular processes, so it's very easy to contaminate because we have all this material on our bodies, so if we're touching the area, it can contaminate. So they have their stuff there. We have basically a chemistry bench where we're going to run some of those dissolved nutrients that Kayla was talking about. We have a centrifuge. This is my little workhorse. It is 20 years old and it is still running. It's kind of like a lab family member, but it comes to sea with me because my new centrifuges I don't want to come to sea. But this is a little workhorse. You might have mentioned Poral Water. I'm getting Poral Water from sediments. You spin it down. It has a test one in it.
Yeah,
Great.
if you want it.
Yeah.
I'd like to see it.
So do you have your before, or do we need to ask if Kendall would grab it out of the...
So
Oh, you got it?
This is the before tube, and you can see it's full. Don't see any liquid solid separation. And then,
Let's see.
well, here's the after.
Ooh.
Okay, so I'm going to put this into a syringe, and I'm going to put a filter on it like Claire did outside, and I'm going to filter it very carefully into a clean sample bottle, and then I'm going to freeze it. I'm going to label it and freeze it.
When she says clean, it's very clean.
Right, so
For trace metals,
Yes.
which is challenging to do, and it takes time and a very rigid protocol to clean things.
yeah, so we've covered everything in our work area with plastic and saran wrap to keep it clean. But sometimes, these are not free of organisms, so sometimes a worm will get caught in our sampling protocol. So that's a frozen worm that happened to be caught in our sediments. That's it.
So other things we have here, pipettes are, they're tools in a scientific laboratory. It's a series of pipettes that dispense a specific volume that you can dial it in. Imagine these like, measuring spoons. You need quick and precise kind of measurements. You can do that. The thing next to it here, this white thing is a peristaltic pump, which allows us to pull water from, whether it's a CTD or something, and push it through a filter. And then if you go over to the left here, we have our MilliQ system to make kind of ultra pure water. And then ultra pure water is kind of what we use to make all of our reagents and chemicals. And this takes water that the ship has desalinated and then has to purify it further. Of course, freezers, fridges, and then this little guy here, it's called a microplate reader. It can do a series of things. You put samples in these plates, which you can see some of the plates on here. Every sample gets read in a well plate, and you can measure things, absorbance, which can tell you about nutrients. You can measure fluorescence, which it's a very nimble. It's a very nimble platform, so you can do a variety of measurements with it.
And can you point out that cast is a little unusual?
Yeah, so this isn't the output that we get from the CTD, and it's in real time or near real time. The instrument's acquiring data really quickly, so like 8 to 24 times a second, depending on the device. Each of these sensors has a color, so the blue is oxygen. When it's dark blue, that's when it goes down. When it's light blue, that's when it came up. So we can visualize how things change between going down and coming up. A lot of the times the boat's drifting. You can see the salinity In the red, you can see this fluorescence that was being discussed. This gives you an indication of the potential biomass of phytoplankton in the water. And then the temperature is a green. You can see not much change in the temperature. This was a pretty shallow site that we sampled this morning.
And can we walk in here and go around into the dry lab?
Sure, go for it.
Okay, we'll just take a peek into the mess hall as we go.
This is the mess hall slash conference room slash arcade slash office slash living room for the whole of the 21 of us here. Ooh. This is Rodney.
Hi, Rodney.
He keeps the boat going. We like Rodney.
Just one of the heads on board. I'm untangling myself from this microphone system.
Who do we have in here?
Ooh.
Oh, we hold the microphone?
Hi. So this is Dr. Chiquita Brooks and Dr. Haviland Steinmuller from LUMCON, that is the Louisiana University's Marine Consortium. They are collaborators on this project. Can y'all explain to us a little bit about the samples that you're taking and what you're doing with them?
Yeah, sure. So some of the samples we're taking, I'll try not to touch the microphone. Some of the samples we're taking are mud subsections, probably similarly to some of the stuff you guys have seen earlier today. And from these mud subsections, we're attaching a piece of equipment called a rhizon, which has a little wick on the end, and it pulls the water down into this tube that has a vacuum. That's what's giving it that pull. And this water down here is what we call Poral Water, so it's water that forms in the pores between sediment particles. And then from there, we're sampling it for dissolved inorganic carbon, which is CO2, the gas, but dissolved, which is one of the greenhouse gases, so things that warm up the atmosphere. Yes?
So before you get more into processing the samples, just really quickly, we just saw a different way of separating pore water and getting pore water samples. Why are you using this technique instead of a different technique?
Oh, that's a good question. So this one takes a little longer to get the pore water, but sometimes we can get a little more volume out, and we also don't risk oxidizing our samples as much, which is kind of a big deal for some of the reduced particle types that we're looking for. So we're also looking for like dissolved iron, which we would want in its reduced form, and hydrogen sulfide, which is another gas that we can oxidize by doing this centrifuging.
So just as a kind of simple explanation of that oxidation and reduction, you mentioned iron. So if you think of iron as like rusting and turning red, that would be the oxidation of iron. So you're using this method to reduce that
The potential for making the rust, yeah. Absolutely, yeah.
As compared with Dr. Ramon's extracting pure water with the centrifuge. She just showed us that in the other lab.
I have to use trace no clean kits. So everything that I use has been acid washed multiple times by my colleagues. So everything is in here has been acid washed, so it has the tube that I put the sediment into. After I centrifuged it, it has a syringe that's also been acid washed, so free of metals, that I would then pour my pour water into. This filter has been acid washed, the sample bottle has been acid washed, so that's also another reason why we would use different methods, because that's important to my work and what I want to know. It's not as important to Chiquita's work, but then we can collaborate and we can share our results. Yep.
So what processing are y'all doing on board? And then can you kind of give us, I don't know, a broad picture of what you'll do on board versus what you're taking back with you?
Yeah, so you caught us at a good time. Dr. Steinmuller is actually running some of that DIC, so the dissolved inorganic carbon, right now. So I will describe to you her process. she's going to stick a needle attached to a syringe into this vial, and then she's pulling the headspace air, so the air over top of this. So we're seeing how much CO2 has actually been off-gassed by the water. And then we inject that gas sample into this little port over here on this machine, which is called a gas chromatograph. And then what our screen is actually as the gas moves through the machine, we'll see a peak here for methane gas. And then this gas chromatograph has what's called a methanizer, so it will reduce the CO2 sample that we put in here and reduce CO2 as methane, so we'll also see a nice little CO2 peak right here. It's going to take about 3 minutes.
So then, just kind of broadly speaking, you can kind of speak to your own samples, but just in general also, if you want to just kind of throw out an idea for how much of this gets processed right here and now versus how much of it gets taken back to the lab and we continue processing them. How long does that take?
We're probably processing like a 20th of the collected on this cruise, so a lot of our samples have been preserved, so we either preserve them in acid or we preserve them in a anaerobic headspace. So for example, these are to measure the methane gas that's in the sediment from the different subsections. And we preserved it. Actually, you preserve the methane in a base, so it's in sodium hydroxide, and that will kill all the microorganisms, so then we can measure the methane in the headspace of this vial and get a sense for where methane versus CO2 is produced in those different subsections of the soil core.
Thank you. So about how much do you expect to process after heading back to shore?
Well, it could take years, and that's not an underestimate. You know, certain things will have priority, but then there's other samples that we hold on to. My students and technicians have called these my emotional support samples, but I don't want to throw them away because once we've characterized a lot of the properties of these sediments, then we have this great data set for people to go back to these samples and run new things. And if the samples have been preserved well, then you can leverage this grand data set that we've collectively made. We have people from multiple institutions here, and it seems like, hey, we have some samples, you have a question, you can come in and you can run them. So immediately we got to do these in probably a year or two to finish off the main of the samples that we're taking, but we'll likely hold on to them for a little longer than that.
And so can...
Oh, sorry, you were asking a question. We
No,
have
go
a
ahead.
little peak.
Oh, let's see.
So here's our CO2 peak right here. And so we'll use this area. And so when you were asking about how long samples are going to take to be analyzed, for me, I'm interested in how CO2 forms as a rate. So that formation is going to happen over time. And I keep them in these slurries so that I can measure how much CO2 is coming out at different time points. So for us, our last time point will be 14 days after we actually put these things in our incubation vials. So we started, we'll start the last set today. So our data collection won't be done until 14 days after today.
Thank you. So can you just kind of do a really broad rewrap of the different kinds of data collection being done by the different groups? So metals, oxygen, microbes, just kind of a broad overview, and kind of put that in scope for what we anticipate learning from
we're
analyzing that data.
Typically we've talked about processes in just the sediments, but we're also looking at processes in the water as well. So really we want to understand on this cruise how a specific element is flowing kind of from the sediments to the water, and as was said earlier, that's important for fueling productivity by phytoplankton called diatoms. But the other thing about silicon, the element that we're studying, is that over geologic time and over longer time scales, it provides this kind of counterbalance, or really important either counterbalance or additive effect to CO2 in the atmosphere. Weathering of silicon from rocks consumes it. Processes that we're studying in the sediments can actually evolve and make CO2. These chemical processes that we're studying. So really, we're trying to link carbon and silicon cycling in marine sediments in this area. And this is a very interesting area for a lot of reasons. Economically in the Gulf it's very important, but kind of ecology and biogeochemically globally, these are really important systems, these river plume systems, for trapping things that come in from land, for making these pretty massive phytoplankton blooms. And big picture, we just want to understand how this whole system works better.
Well, thank you so much, all of you, for taking the time to talk to us and tell us a little bit about the science that's being done on the qualified cruise. And those of you who joined us, thank you for tuning in.